I'd like to introduce Dr. Joe Orsini. Joe is the Director of Operations at the New York State Newborn Screening Program, and he also is the supervisor of the Lysosomal Storage Disease Laboratory.
Thanks, Beth. Not sure if I'm close enough. Is that good in the back? Okay. So we're going to go over today New York's assay, which is a mass spec assay. I did not put in here anything about a fluorescence assay or what Missouri is using, so this is really just covering one type of mass spec assay. I believe there'll be another version of it covered. But there are fluorescent assays available, as well as this digital microfluidics assay that Missouri is using. I don't claim to have I don't have any experience with those assays, but we're going to just delve into this mass spec assay. Go over the testing algorithm, and then I'm going to go over screening data for Pompe. We've been screening for a year, a little over a year. We started October of 2014, and we've been pilot screening now since March of 2013 for hurlers. So that's on a subset of New York State's population. It's only out of New York City hospitals. There are 4 hospitals participating, and it's a consented study. So there's a lot less information we have on that screening thus far, because it's about 500 samples a week. All right. Gotta make this thing work. I don't see that. I did, and it didn't go. All right, there we go. Thank you. So we started screening with the CDC reagents. I don't know if any of you are aware of that, but we made a change this May, June, and the beauty of this, the change was that now all of the solutions are much easier to put, all the reagents are much easier to put in solution. We get to formulate the size vials and the amount of material we want to run. So what used to be running with, say, 50 vials worth of solutions to get a month's worth of reagents, now we have 1 vial a day. So each day, basically a working day, is in a vial. And so we can run 15, our plate number is about 15 plates a day. So that adds up to about 250,000 patients a year, which is our birth rate. So we did switch, and it's now the available reagents are from PerkinElmer. I believe there's still CDC reagents out there, but eventually those are going to be no longer provided. So we basically made the switch quickly just because we knew it was coming. There are, like I said, some advantages for the lab as well. Then one thing we did have, just to put up some notes, daily mean did drop for Pompe from 15 down to 10 micromole per liter per hour. IDUA has been at 7.2. We didn't have any experience with IDUA with the CDC reagents, but that change is largely because we're trying, you can run 6 LSDs with this assay. The assay we had originally set up with PerkinElmer was more optimized for 4 that we were looking for at the time. So this slide shows the actual substrate internal standard pairs, or primarily the substrates that are used for both Pompe on the top and IDUA on the bottom. The interesting thing to me is, to the average person, you'd look at them and say they look the same, but there are some minor differences, and certainly the enzymes can tell the difference. If you look at the, can't see the arrow. This carboxylic acid group down here is largely distinguishing. So both are on a glucose frame, and the enzymes are cleaving the sugar moiety or sugar-like moiety off in both cases. So you can see this is the enzymatic product for Pompe. Oops, now it's going to go crazy on me. Now you know my whole talk. Sorry about that. So you've got the enzymatic products on the right, but you can see the molecular weight differences, and the mass specs can certainly tell the difference between. What you see are the deuterated versions all around the benzene ring, and that's what helps with this assay tremendously. Compared with the old assay, every substrate or internal standard actually has a deuterated derivative of the enzymatic product, so it's very easy to get accurate quantitation. We see across instruments, we see much better stability in the assay, as well as kind of the daily monitoring of the mean, which is something we track with these assays. The bottom of the slide is just an example. You've got your wish I had a pointer. But you have your, there it is again, the substrate interacting with enzyme in the blood, forming your enzymatic product with a free sugar moiety. So this is our assay. It's been the assay we've primarily used all along. Some things have changed since we started screening. We started screening for Krabbe disease almost 10 years ago. This assay is different because the reagents are different, but some of the backbone's the same. First thing is 19-hour incubation. This was a requirement when we started running for Krabbe because the enzyme it's the bad actor of the group. The good thing about working with the bad actor of all the LSDs is you get to learn from the school of hard knocks. And Pompe seemed easy coming into that. MPS1 is somewhere in the middle, and you'll see we don't have as much experience to say how it's going to be, but it seems to be more like Krabbe in the sense with the number of false positives. So the incubation for overnight, 19 hours. Then we do a liquid-liquid extraction, dry the plates, and run on a mass spec. We removed an SPE step we had temporarily in place when we were running this assay. When we first started, we didn't require it, but when we went to adding Pompe and MPS1, we figured we'd have to bite the bullet. But these new reagents, they're a little more volatile in the mass specs, and because the mass specs have gotten more stable or insensitive, we're able to put a lot less sample into the mass specs. In all cases, all of our testing, once we have an activity, we compare to the daily mean activity and use a % of mean cutoff. That I felt was required in the early years where we didn't really know the stability of the assay. You could probably get away with just using a target activity once you develop a mean for your enzymes. But we've been sticking with % for now. Okay, so here's our algorithm. It's going to look very similar to every time that anybody may have seen a Krabbe algorithm. We test all the enzymes. If it's less than 20% of the daily mean, and that's a %, it's not a percentile for results. It's actually just a direct % of the mean. And for Pompe, that amounts to about 2 samples every 1,000 or 1 sample every 1,000. So it's a really small number that hit this bottom rate. For IDUA, it'll probably be more like 5 per 1,000. In Krabbe, it's somewhere in the 5 to 10 range. We go ahead and do a duplicate or triplicate testing. Sometimes we do more testing depending on the quality of the blood. If the activity is less than 15% for GAA or IDUA, then that is considered a screen positive by our first tier test. We're fortunate to have a molecular lab. We run DNA testing and for both Pompe and for Krabbe, but IDUA, we're not currently doing that. That's being done at the Mount Sinai at the center there. So those will be anything with 1 or more mutations is screen positive. If it's above 15%, screen negative, or if no mutations, screen negative. This slide shows a sample exchange between Missouri and New York, and I like to bring this up because it was a blinded sample. Or a blinded study. We had 38 samples come in. We weren't really sure where we were setting our cutoff. We had set it at 15%. And you can see as you go down the slide from the top right down to the bottom right that's increasing activity out of our lab. So the lowest activity samples are at the top, and the bottom is the highest activities. And you can see that the classic infantile and non-classic infantile cases are all under 6%, 8%. The lowest, I think, is 5.1 there at the classic infantile. Then you start to see some late onset sample activities coming in. And then as you go up, genotypes of unknown significance and pseudo-deficiency in carriers. This trend is something we've seen in our screening as well. And I think, the advantage of the 18-hour, and that's why I pointed out, our 19-hour incubation, is you start to get a little bit of a spread between the turtles and the real slow turtles and the slow turtles. So at least that's my way of explaining it. It's very easy to see things bias low in an enzyme assay. So the fact that we see a late onset with really low activity, it's alarming when we see a low activity sample, but it's not something we necessarily say it's got to be an infantile case. We look at all the other enzymes that we can, and for our state, that's only sometimes we're lucky and it overlaps with one of the pilot studies, and we can get a little more information. But we are looking at actually retesting our samples on a full panel LSD list, because you get and call them reference enzymes rather than actually label them as LSDs that we know they are, mostly just to keep ourselves fooled from looking too closely. But once we get into where one of these enzymes low, if others are low, it's always the same. From our experience, has been a sample quality problem, or at least that's the thing you start to question first. So here's our population study stats for crab A. You can see under 15%. It's the bottom section of this table of things that have actually ended up going to DNA. 112 samples in roughly 260,000 specimens that we've tested thus far. So I'm not going to. You can see that the numbers go down quite a bit as you go up the table to where less than 7%. We've only seen 11 samples, 8%, 12 samples. So the number of really low activity samples with this test is pretty small. Now let's take a closer look. This is where we're looking at all the samples that were under 15%. So would have after retesting. So that's 62 of them that we've ended up doing DNA testing on. Of those, we've referred 48 infants, and I'm happy to say up until 2 weeks ago, it was like, where's the infantile case? New York's been screening a year. We had not seen a single infantile case. So we did detect one. It was a couple weeks ago. That child was a 35-week premature baby. So I'll tell you a little more on that story, but you'll see down at the bottom some of our premature babies. We see a fair number of false positives in the premature. So, yes? How long does it take to get the DNA back? A couple days. Yeah, we're really fortunate. We have somebody pretty dedicated to it because of some funding we have on right now. So not quite as fast as crab A. The interpretation turns out to be fairly extensive for Pompe, and there'll be a little more explanation of that, but not much later on in the talk. But the first case, I'll point out activity at 6.1%, and I hate to be happy about this. I'm happy we finally detected one, but this child did have cardiac involvement and is CRIM positive. So fortunately, the child's actually going to be put on, I found out just a few minutes ago before this talk, going to be put on ERT starting tomorrow. So very young and early to get this child started. So we're really happy we finally have detected an infantile case. For whatever reason, New York seems to not get a lot of, I mean, we've been screening for crab A for 8 years and have 5 cases. So I don't know if it's something with our mixed gene pool, but anyway, that's the way it's been working. So then we have this other group, and I hate to call them late onset cases. I've been calling them potential late onset. We've come up with different names for them, probable, possible, but to me they're all potential. None of them are real cases yet, and that's one of the reasons I asked the question of Tony earlier, was whether any of the late onsets have presented yet, and the fact that they haven't puts a lot of what I'm going to say is the thought-provoking part of this talk, which is, you know, especially in light of when you look at cutoffs and our cutoff, I wanted to go back to our algorithm here. So we had set our cutoff at 15%, and I think I showed you earlier, or this slide, where the infantile and classic cases are in the 8% or under range. Then there's some late onsets, and you start rolling into these genotypes of unknown significance. And as I stated earlier, that's a lot of what we're seeing in our population. And I think conservativeness always leads us to have our cutoffs be higher. That's what got us started at it. We end up in some of these circular arguments because when we were setting our cutoff, and we said, well, we're going to stick with 15% because there's a 12.5% here in the late onset from Missouri. But so far, that's not a real case. So it's all based on the fact that it's got low enzyme activity, but not real low, not low enough that you would call it an infantile case, and genotypes that are more associated with later onset disease. So we're running into tricky territory with this, and it's still, there's a little more to the story on that when I get to come back to this slide. So coming back, we have these 15 potential late onsets, and I have up there the range of activities being 5.8 to 10.8, and I actually meant to update that. It's now 14.2%, and we had met as a group. A group of us met with the New York State IMD directors. A couple of them are here, and we had said, okay, we're going to change the cutoff to 13%. And I got back to the lab that week, and we had one that was cooking. And ended up we got diagnostic information back that this was another potential late onset case. So I was like, okay, 14% for anybody? Well, 2 weeks later, we have one at 14.2%. So it started at 13, one at 13, another one 14. So here we are, stuck at 15. And I don't mean to, I wish I had a crystal ball, and I think we all wish we had a crystal ball, but we don't know who of these are going to present and win. And when you start looking at across literature on people reporting incidence of disease for LSDs, it's always based on low activity and mutation analysis. And the mutation analysis, we all know you can't necessarily predict who's going to end up with disease. Some of our late onset patients actually have this splice site mutation. It's in their homozygotes for a splice site mutation for Pompe. And for experts in the field, and I'm not one, but I'll talk about it a little bit anyway, it turns out that studies are showing that people have that but don't develop disease, and there's a fair number of people that won't develop disease with those splice site, as homozygotes with that splice site mutation. So part of the problem is now diagnostic labs aren't really haven't been completely set up to be able to handle a question, and is enzyme going to be able to help us more in the future with predicting these? That'll be a question that I think we need to look at over time. Okay, so of the infants not referred, you see 8 with pseudodeficiency alleles only, ranging from 9.4 to 14.7. So my point was really going to be that most of them are on the higher end of the scale. When we ran real control samples from known patients with infantile, we've found everything under 8%. About 10 of them. I don't have that data up here or anywhere else, but that's what got us started on this idea. Well, we'll go with 15% to meet these late onset cases, but we don't even know they're really going to be late onset cases. So then the next thing, 16 infants with normal allelic variance between 11.5 and 14.7%. And I want to point out that this is where all but one of them were NICU or premature infants. So I think Missouri will say the same thing. They see a lot of false positives in the premature group. So the cautionary tale is we have our infantile case was in the premature group. So and had cardiac involvement and is going to be going through ERT. So we're really quite happy that we have molecular on that to be able to give us guidance. Oh, that's okay. We don't, and we're looking at it, but I'm actually a little nervous about doing it. I think it's to set those kind of cutoffs. I know Missouri does. Because we have the molecular and we can just jump through that hoop, it benefits us to get to an answer faster. But it does mean that there'll be cases that we're going to refer where we find carriers, and because it's a kind of a double-edged sword. But you can see, though, we have saved a fair number of referrals, 8 with the pseudodeficiency alleles and 16 with the normal allelic variants. And by those, they're in the database as being variants that knock down activity, but not thought to be significant enough to be disease-causing. And by that I mean the GAA database, and I'm not sure what it's called right now off the top of my head. I don't get to do that part, thankfully. So this is just some of the slides showing infantile and probable/possible late onset cases. And I'm going to stick with possible. I'm not really sure about the probable, it's just the way it's been labeled. As I was stating earlier, I don't know if you can see very well, but a lot of these are the red mutations are splice site mutations in almost all of our late onset cases. There are a couple of, there's one very common splice site mutation and one less common one that we found. So they're in amongst our lower activity range. Ours is, this is screening labs activity for percent read. This is screening lab activity. And we were getting all of our data going to Mount Sinai for the diagnostic follow-up lab testing. So these are, you can see the numbers at the diagnostic lab. There's one not done, one pending. They're on the lower side. I don't know if there's any difference between these, but our infantile at 3.1 there is some separation in these activities, and whether that will amount to something that helps us in the future, only time will tell. So just the numbers here, 48 referred cases and 260,000 births. So that gives us a screen positive rate of about 1 in 3,600, 0.028%. Our referral rate, so I like to think of that positive rate as if we didn't have molecular testing, where would we be? So you can help people who are setting up without molecular to gauge what a mass spec's positive rate could be, at least with this set of reagents. The referral rate being a fair amount lower, at 1 in 5,400. We have this potential late onset incidence, 15 late onset cases and 260,000 is 1 in 17,000. So that's a fair amount higher than what's been reported in the literature, and begs the question of, are we actually finding cases that would not be observed otherwise, or is our rate that much higher because we're finding cases that won't be cases? So that's what I was talking about earlier, the conservative side of me of trying to make sure we catch all these late onset cases when you don't know really what they are. And it's difficult for us to go back and get control specimens from late onset individuals when they were a baby. So it's possible to go to a screening lab. Some labs have archived data or samples, but those samples are always going to be aged, and age in time wither away enzyme activity. So you lose a little bit of the ability to really tell what's going on. So that's the quagmire or dilemma.
I mean, I think the question we've sort of been thinking around on some of your slides is, what is the target of your screening assay? Is it early infantile? Is it all possible? Like, what was decided by New York that this is what we're going to try? Because it looks like it's any form of Pompe we could pick up. And I think that if you think about other disorders, Krabbe is one where hindsight is wonderfully 20/20, and you can adjust. And we do this routinely now with even galactosemia to try to hone that. You want to look for classic, it's only classic. If we don't have a DG, so be it.
Mm-hmm.
So could that help benefit from your other disorders hindsight for the foresight of Pompe?
It would guide us if we had some of, I guess late onset. It really, to me, begs the question of what late onset are we looking for? I think everybody here, I'd like to maybe an unofficial poll. Who here would like to see us detect children under 5 years of age? I think I would, I don't see all the hands there, but I'm going to say that most people might want that. But certainly the infantile and the non-classic infantile, 2 years old, what about 10 years old, 20 years old? And some of these, especially the mutations with the two splice site, slip-free mutations, those people generally aren't presenting until adulthood. So, and if they'll present. So is that an indication of, are those activities an indication, and we don't have, of where our cutoff could be to avoid them, or do we really, is it worthwhile to catch them? And it really falls into a category of, I feel like it's above all our pay grades to make this decision.
Where they're 5 or 6 years old, when you can go back to their newborn screening times and take a look at them.
Not a lot, no. Yeah, that's part of the problem, and we're trying to assess. We try to get those, but No subtalk.
Those of us who were at the Pompe meeting that the State Lab sponsored a month ago, Krishnan showed a video of a child who was homozygous for this white mutation. And she showed it specifically because I follow one of the patients who was identified with this mutation who actually had an abnormal PK.
Mm-hmm.
And what she showed on the video was a loss in motor ability. And was jumping on the video, and then she stopped it and it was as well. And so this was an example of somebody who was, I can't remember the age, but 10 or 11 years of age, who has homozygous with this very mutation.
Right.
So I think it raises this, I think these are all good questions. Who are we targeting? And we also have to be aware of the fact that, I think that there are a lot of things that are known, which is probably why we passed this like that. But I just want to make sure I had it sort of research that.
Well, I'm not certain. I think there are some, there are, the youngest is maybe in that range, but the vast, majority are 40s, but, 20s, 40s, adulthood. But again, what is to me, it's more relevant of what range. I don't feel like I can make that decision of what age to choose, but I know we could nail the infantile cases, at least eliminate a lot of these potential late onsets, but do we really want to do that?
You said to me that with late onset Pompe, we don't know if they're ever going to become symptomatic, but we were pretty sure with late onset Pompe that they would become symptomatic.
Well, I don't know if I said that, but I know other people said that. So that's the dilemma. I don't feel like I have the expertise to say that at all. I think part of what we're going to learn through newborn screening is just that. But with everybody, it's going to be, we don't necessarily have the setup to monitor fully. We have to be this is a lesson from the Crab A world. How do we monitor the patients in a way that keeps them wanting to come to follow-up and doesn't scare them, isn't so onerous that they never come in, that they give up and they just feel like they've been beaten by the medical system. And I don't know the answer to that, but I think that's some of the critical questions we need to answer ourselves. And I don't know even what the form of that is, but I don't want it to weigh on just my shoulders. I can magically change the cutoff, but is it something we should do? And I felt comfortable with moving away from 15 and down to 13, but now I'm not so sure. And once you find or detect somebody that's potentially going to have disease, then you feel like you're kind of stuck. So it's almost like, in this case, should have we done it the other way? Started to just catch infantile and go on from there. Anyway, I'm going to make a quick switch to Hurler. I don't know how my time's doing, but Hurler, the enzyme structure has been known for quite a while. It's also known with Pompe. I just threw that up there so that I'd have something interesting to look at. But that yellow section is the so-called barrel where the active site is. And it'd be nice if more modeling would actually work with these. And I've seen that there's some potential for that where you have point mutations that may be on the outside of this protein versus in closer to where it might affect the active site. So those kinds of things may offer some hope for prediction, but at this point it's not really working that way. Certainly missense or nonsense mutations have been associated with disease, but the missense are the ones that are more tricky to contend with. So this is a slide with a bunch of positives. We had to validate our current assay, and you won't see GAA or Pompe up here, but there is IDUA. There's only 2 controls. And both of those are in the light blue, kind of in the middle of the slide, with a percent of daily mean at 1 and another one at, I think it's 2.2. It's a little hard for me to see from here. So giving you an idea, we're kind of in the same boat. 15% is our cutoff, but we've got some real cases that are very low. but these are older blood spots in our archives, so we always have to take it with a grain of salt. I learned a really important lesson is don't trust those blood spots that have been sitting around, and don't trust activities from older patients. If you get a sample, if you're setting up your screening lab and it's from an older patient, then their blood white cell counts at birth are a lot higher, about double that of an adult, maybe even 4 times or more of that of an adult. So you got to be cautious with taking samples and controls from anybody other than a true newborn. Even an older infant isn't the same as a newborn sample. So this is another sample exchange with Missouri, so I thank them again for this, for the ability to do this. It's a big help. Oh, I'm sorry, I jumped the gun. Oh, yeah, sorry. This was just to give you an idea of when we look at 20, this is 5,000 samples screened, all of them that are under 20%. And I should have worn my eyeglasses, I guess. You can see that there's a fair number of IDUAs. This is the column we're considering. I mainly wanted to show you that out of 5,000, we have about 20 with less than 20%. Remember, that's our first cutoff. So that for us is 5 days worth of testing. So divide that by 5, and that's be our retest list kind of on a daily basis. But the main reason I wanted to show you this, if you look at the bottom, you got low ABG. This is in multiplex testing, so we're running multiple enzymes. You can see IDUA is low. Very often ABG, which is Gaucher enzyme, is low as well. These two track together. So if you're multiplexing, luckily to have all these enzymes all the time, one of the things we've observed is ABG and IDUA somewhat track together. And if you're doing multiplexing, GLA, which is the Fabry enzyme, and GAL-C track together. So you get a lot of valuable information, and we're actually going to work with Minnesota and Piero Ronaldo and his group and load our data in, because I think into a database, and over time there'll be a better way of predicting real positives from this, and something that you can look at and get a feel for. It's obvious when a sample looks like this, 4, 7, 12, 19, all low percent. That's a bad sample. So, but it's not so obvious when you get them where they're in their 40s and 50s, and how should you gauge that? And we try to gauge our activities along that line when we get to do multiplex, and that's one of the reasons we are looking to implement it as reference enzymes for everything we do. All right, so here's the sample exchange, and you can see we only had one hurler case in amongst the group. But once again, these are all, there was some normal samples that were sent to us, as well as pseudodeficiency samples. And you can see in this case, the pseudodeficiencies are hitting our 15% cutoff more often than they were with Pompe, so maybe 15% is a bit high. We got hurler at 6.1, but then there's some carriers at 2 and 3. I didn't have the advantage, I don't remember what the other enzymes looked like for this, but so for whatever reason, carriers are very low, and it was just probably a sample quality issue, I'm not sure. But again, hurler at 6.1. Okay, so now we're up to 5,800 samples tested, and our referral rate would be 1 in 1,900 based on the fact that we have 3 referred cases, for 0.018%. So it seems to be at about, that's our CRAB-A referral rate, so just to give you an idea. And it turns out our CRAB-A activity is right around 6 now, 6.5. So MPS 1 is probably going to behave somewhat similarly. But because we aren't doing genotyping, we aren't able to pick up whether there's pseudodeficiencies or not in this group until later. So then we have, of those 3 referrals, we have one at 12.1%, one at 11.5%, and one at 13.5%. So all 3 of them so far were on the higher side of our cutoff range. We're still awaiting diagnostic lab results on the 3rd one. It was just referred 2 weeks ago. But the first 2 had, the infants had 2 variants of unknown significance, and the activity was not in the affected range at the diagnostic lab. We do have a sample that's in the cooker now at 4%, so we'll see what that one is, if that might be our first case. And the other enzymes, since we're running at least 3 other enzymes, we're in the more normal side for that. Our false positive rate 2 is 0.034. I mean, all of them are false positives to our knowledge at this point. Out of the first 2, we're still waiting on the third. So 2 in 5,800. We'll see how the rest goes. And that's my talk. I'd like to acknowledge my group, which is Monica, Chad, and Ryan. They're the ones running the assay, and Michelle for guidance, and then Colleen and Aaron do a lot of the all of the molecular interpretation and run the tests, and then Chun-Li and Melissa from Mount Sinai for diagnostic testing and help with the pilot study. And Priya, Diksha, they're at Duke, and they've been looking at all of our cases. We present them go over the cases on a, say, monthly or every other month kind of a review of the cases, because they're very familiar with the mutation analysis. And then the folks from Missouri for sharing their samples, Patrick, Carlene, and Tracy, and Wei, Bob from CDC for all the controls they provided us. Any questions?
When you're talking about activity, percent activity of the meat, are you talking about functional activity or enzyme level activity?
Enzyme level, and it's strictly based on our lab's assay. So if the daily mean is, I'll use the best example, Pompe is 10, and if the baby has an activity of 1, it's 10% of the daily mean.
So make sure that it's not that idea about multiplexing. I mean, do you check other enzymes are low as well on the same system?
We only routinely are running Crab-A and Pompe because that's what all our states mandated to do. So we don't. It's not enough information. Typically, if both of them were under 20%, then we do call it a quality problem. So and just wait for or request a repeat. Trouble with that is often we end up in a case where we get another repeat and it's bad, and it's another repeat and it's bad. So most of the time they do elevate. So we're hoping to go to the other ability to look at all the enzymes and really have a way of evaluating them against each other. I think that'll be very helpful. Yes, yeah. The repeat specimens often, well, there's 2 things that happen. Often the repeat sample, the quality isn't quite as good. It's not always taken at the hospitals where they're used to doing it. It might be taken at a private physician's office. And then the fact that they're older, their white cell counts have dropped, so we almost always see a little diminish in activity. Any other questions? Yeah, sure.
Any problem with the blood transfusion? Are you looking at what? Do you look at the white? Do you have anything to do with the white blood cells?
Well, we do not know at this point whether a baby's been transfused. We get transfusion marked on our card, and the presumption is most of them are red cell or platelet transfusions, but we don't distinguish between those and white cell transfusions. That's something we're looking at adding to our card, but it's always a process to change the card, a big process. So, any other questions? Thank you.